Right, well, good afternoon, good morning, good evening, wherever you are around the world. You're very welcome to join us for this PGC Worldwide Lab meeting. And this session, we're delighted to have three speakers talking about the bioinformatic approaches that will be valuable for many different groups as they explore this, this wealth of genomic data, genome-wide association study results, and we figure out what we can do with all this richness of data to start making sense of it. So I'm Cathryn Lewis, I'm Professor of Statistical Genetics at King's College London. I lead the PGC MDD working group along with Andrew McIntosh, and I also take responsibility for education and training in the PGC. And we have three speakers that you can see on the screen, and they're going to speak in the order of Oliver Payne first, and then Patrick Sullivan, UNC, and Michael Love, also from UNC. So buckle up, and we look forward to a really exciting hour. A couple of practicalities. We're going to run the talks one after another, and then take Q&A at the end. You'll see in the center at the bottom of your screen a Q&A button, and you can write your questions there while you remember listening to the talks. And then at the end, you'll also have the opportunity to put up your hand and ask on screen if you would rather do that. So without further ado, I'm going to hand over to our first speaker, who is one of my close colleagues at King's College London and is always a delight to work with. So Oliver Payne is a Sir Henry Wellcome postdoctoral fellow with us at King's College London, and he's going to talk about GenoPred, which is his comprehensive and scalable pipeline for polygenic scoring, which we've really enjoyed using internally, and it's great to share that more widely with the community. Ollie, I'll hand over to you.
Great, thank you, Cathryn. Hello, everyone. So as Cathryn said, I'm going to be presenting this pipeline called GenoPred for polygenic scoring. So brief introduction, as I think most of us agree, genetic variation is important in determining individual differences in a broad range of health-related outcomes. And in the past couple of decades, huge progress has been made through genome-wide association studies in identifying robust genetic associations with a broad range of outcomes. One of the key applications of genome-wide association studies is polygenic scoring. So a polygenic score summarizes an individual's genetic risk or propensity for a given outcome, and they're typically calculated as the GWAS effect size weighted sum of risk variance that individual carries. Now, these polygenic scores have been used extensively in research to answer a broad range of research questions, such as the genetic architecture of traits and the overlap between them. And recently, they're emerging now as a tool for enhancing personalized medicine as well. So quite an exciting point. So you take an individual's genetic data, you perform this polygenic scoring process, and that allows you to place them on a kind of continuum of genetic risk for that outcome. Now, the process in the middle of the screen, the polygenic scoring part, can be quite complicated. It's rapidly evolving in terms of methodology, and there's still limitations to certain aspects of that. So we've developed GenoPred. So GenoPred is a research program that we run at King's College London, and we're looking at a range of different aspects of the polygenic scoring process. I'm not going to go into each of these studies now, but look them up on our website, which I'll put a link on at the end if you're interested. But I think, throughout all of these studies, some of which published, some are still ongoing, we've always made a special effort to make the code we've used publicly available so other people can apply some of the tools we use to kind of calculate these polygenic scores. But, uploading code online is not always very accessible for a lot of people, especially if they're not familiar with these types of often large genetic data sets. So we developed the GenoPred pipeline. So this is a software pipeline that aims to kind of make the process very easy with a few kind of simple inputs, then with options for people to choose what they want to happen, and then a kind of standardized output that people can then use to access the polygenic scores and other information. So it's very easy to use. It implements a range of leading methodology, which we aim to keep up to date to integrate kind of new methodology as it comes out. Importantly, it calculates reference standardized polygenic scores. And this is really important. I'll give a quick insight into that. So if you're going to use a variable in a prediction model, it's really important that that variable can be derived in a future target data set in the same way it was in the original training data. And the pipeline does this by using a common reference data set to generate the polygenic scores at various points. The pipeline is robust. It's been used extensively by me, other people at King's, and also external collaborators. And it's consistently providing reliable outputs. And it's also highly scalable. So you can use this to analyze many different GWAS data sets and also very large target samples. So to give a bit of an insight into what's going on under the hood, it's a snakemate pipeline. And this is really useful because it ensures that the outputs of the pipeline have come from the inputs that you think you've provided. And it does this by checking the timestamps of the files, but also checking if any code has changed, that maybe steps need to be rerun. And that means you can kind of go back and forth, make updates, and the output is always going to be reproducible. It's also highly scalable, and I'll talk about that a little bit more in a moment. It uses Conda software environments. So the software that's used by the pipeline is kind of very easy to install, and it will always install the same versions, improving reproducibility and also avoiding kind of complicated setup, which is often installing the software is challenging, but this resolves that. All of the code is publicly available on GitHub. It's under version control. And importantly, we've developed a range of documentation describing how to use the pipeline. And I'm just going to give a quick demonstration of that. So this is the GenoPred website. And you can go to the pipeline page where there's a kind of promo video, but also links to the instructions. So this it provides detailed instructions on how to use the pipeline. We've also developed a range of explainer videos that people can follow if they're not familiar with using this type of software before. You can follow these instructions. There's also extensive kind of technical documentation so that once you've used the pipeline to perform your research, you can find the information you need to then write it up. So I'm going to talk a little bit about how to use the pipeline. So it has just a few simple inputs. First of all, the config file, and this is a file telling the pipeline what you want it to do. The next file is the target list, which is a file listing which target data sets you want to calculate polygenic scores in. And the other is a GWAS list, and this is a file indicating which GWAS summary statistics you want to be used to generate the polygenic scores. So this is an example configuration of the pipeline. So the config files at the top has a few simple parameters such as where you want the output of the pipeline to go, what is the location of this config file itself, what is the location of the GWAS list file, the target list, and then you can select a range of polygenic scoring methods you want to be applied. Then the GWAS list, each row is a set of GWAS summary statistics that you just provide a few simple bits of information, such as an ID for the GWAS that you can make up, then the path of the GWAS summary statistics file, then a few other pieces of information describing the sample used to derive the the GWAS summary statistics, which can usually be easily found. And importantly, GWAS summary statistics formats vary widely, and this is often a bit of a barrier for people using them. But we've used a script to kind of interpret column names as best it can at allowing a range of formats, but also it'll resolve kind of common things that are kind of hurdles, such as detecting genome build, inserting RSIDs, checking that the frequencies match up and there hasn't been an allele kind of swap and things like that. So generally quite easy to use. Then the target list, similar format, except each row is a different target data set. Again, you just provide a few bits of information. You give it a name, you tell it what the path of the genetic data is, and then you also tell it what format that data is in. It can either be in Plink1 format, Plink2, VCF, BGen, or 23andMe. And then this final column indicates whether or not you want the pipeline to generate reports summarizing the results for each individual within each target data set. And if you're working with very large data sets, this probably isn't what you want to be doing. But if you're just looking at one person's data, then this could be useful. So as I said, it's a StateMake pipeline. Therefore, you use a StateMake command to run it. I'm not going to go through all the parts of this, just to highlight this final kind of command that you give StateMake indicates what you want the pipeline to do. And here I'm using the output all command, but there's a range of other commands that can be used to generate different outputs. So this is a schematic showing all of the different steps of the pipeline, but also showing what command is required to generate which of the outputs. So I'm just going to zoom in here to give an example. If you want to just perform quality control of the GWAS summary statistics, then you can look at this graph. You can see, okay, here's the quality control GWAS sum stats. So I need to use the sum stat prep rule. So I just write StateMake sum stat prep, and that would do it. I mentioned the output all rule. This rule is commonly kind of useful. It essentially runs most parts of the pipeline. So this is the command here, and it will trigger everything it requires to generate that final output. So I said it's very easy to, it's very scalable, and this is, in part due to StateMake's kind of back end. And so you can change it to run on just a single core using this J parameter. Or if you have multiple cores, you can change it to use the number of cores you want to use, so it can run things in parallel. Or if you have access to an HPC or a cloud computing server, you can easily leverage that. And that means you can run, hundreds of kind of jobs simultaneously, making it very efficient. Just as an example, I ran the full pipeline using 14 GWAS and all 6 polygenic scoring methods for UK Biobank, which is 500,000 individuals, across 50 cores, and it took about 6 hours. So very, manageable kind of time. The output of the pipeline is clearly documented and kind of easy to navigate, to find different information, such as ancestry inference results, the polygenic scores, the reports of the pipeline. I'm not going to go through it in detail now. But I've also created a series of R functions that will read in kind of the most key outputs of the pipeline directly into R. So you can, if you want to read in the polygenic scores for a given GWAS in a given target sample, you can just use the read PGS function and specify a few options, and it finds the relevant files and reads them into a nice format that you are familiar with. So the pipeline creates different different reports. There's the individual level report, as I mentioned, summarizing results for each individual. And then there's also the sample level report summarizing results for whole samples of individuals. I'm not going to show you these now for the sake of time, but they summarize kind of quality control information to check that things are looking as you expect. And they also summarize key results, such as the ancestry inference results and also the polygenic scores. And if you're interested, check out those maybe on the website. So I'll wrap up there. A massive thank you to everyone in Cathryn Lewis and also Cathryn Lewis's team. And particular thanks to Miriam and Chris for their feedback on the pipeline so far. And also a huge thank you to my fellowship team, Ammar Al-Jalabi and colleagues, and also my research sponsors and the funders, of course. If you want any more information on it, obviously feel free to ask in the Q&A, but also check out the website. Thank you.
Great, thank you very much, Ollie, for that very clear, very precise overview of GenoPred. I'm sure there'll be a lot of people checking it out. And I can see we've already got one question in the Q&A, but we're going to move on to the next talk as arranged, and it's a real pleasure to welcome Patrick Sullivan to talk on this, and it was his suggestion that we had a bioinformatics focused worldwide lab meeting. And I think this is a side of Pat that we don't always see. We hear him leading the PGC and talking about high-level things, and of course, it is not at all surprised that he still gets his hands dirty with data and does really impressive on-the-spot work. And that's much noted as a real achievement by the rest of us. So without further ado, Pat, I'm going to hand over to you.
Thank you, Cathryn. I appreciate the kind introduction. Today, I'll be talking about a data frame called GeneMatrix. Basically, GeneMatrix, I've been working on this for 10 plus years, and it's pretty mature at this point. The motivation, though, is to combine into a single file the core information that you might need to understand the results of a psychiatric genetic study. It, of course, can be used for many other things too, but this is one that I think I've made it because I was tired of looking at 20 different places to find the information I need. So it's meant to be one data frame. It's meant to be reproducible. Everything is with code, and it's updatable as information progresses. And I've tried to basically adopt sort of a tidy data approach. If you are of short attention span, the too long didn't read version is this. Go to Figshare, search for GeneMatrix, download genematrix.tsv.gz, this file here. Download the README and read the README, please. I update it periodically. The most recent is based on GenCode v45 from 2 months ago. Tad delimited string, things are quote. Well, actually, this is just pretty straightforward. And if you'd like, this shows you how to actually download the file with code and then to read it into R. Okay, one of the key decisions you have to make in this thing is what a gene is, who's going to define it, where it is, et cetera. Multiple alternatives out there. The one that I think is far and away the best is GenCode. Other people can make different choices, that's fine, but this is the one that I have made. After trying to wrangle UCSC for years, I got bored, and GenCode was just amazingly easier, mainly because it's tidy. It's active, it's updated, and I think it's a good source. The URL is there. I use something called the ENSGID, the Ensemble Gene ID, as the stable, unique identifier over time. Its format is given here. It also has a version after it, if you're interested in this. And then I include in GeneMatrix multiple other IDs. Gene name, so you can see this down here. This is the Ensemble Gene ID. This is the main one. I include gene name, the Entrez ID if it has it, HGNC ID. If there's a one-to-one mouse homolog, its Ensemble Gene ID is given, as well as if there's I pick one transcript per gene for protein coding genes, and that specific transcript is listed there. And then it has the standard description. So the GenCode, this is from their page. This is v45, which was released 2 months ago. But as you can see, they go back to 2014. The most recent HG38 version was in August of 2014. Okay, the next couple of slides will actually cover some of the definitions of the 100 columns. All of this is in the README. I use HG38, but I include a liftover for HG19. So the first grouping of data are the basic gene ID, so the ensemble gene ID, gene name. If it's a duplicated gene name, don't trust these. The gene type, the biotype, which I'll get into a second, Is the gene in PAR1 or 2, the pseudo-autosomal regions on the distal tips, chromosome X and Y? Is it in the extended MHC? And then you've got these four variables. HG38H0 gives the chromosome, the gene start, the gene end, and the strand. And then you have the same versions for HG19. There are 38 biotypes in this, and I would argue that we are almost exclusively interested in the protein coding biotype. There are many types. Link RNAs, of course, are important in some instances, but most of these are novel and of unknown function. Processed pseudogenes, unprocessed pseudogenes are probably expert level stuff at this point. There's other types of RNAs, the microRNAs in particular, but I'm not sure this is a very complete catalog. So, and then these are the rest, and some of these are extremely rare. Many of these are one. There's even a category for artifact. So generally, when I do work, I actually focus on I use the filter gene type equals protein coding, as given here. So generally, protein coding is what I work with. Next set of things are evolutionary constraints. So I've got the ones that we created here in the Xenomia paper. I've included the ones from the Nomad group, LOUF, as well as PLI, and a more recent paper that was in Cell looking at the probability that a gene is tolerant or intolerant to haploinsufficiency or deletion, or if it's sensitive to triple sensitivity. So this gives you an idea as to whether a gene is something that may or may not tolerate being a CNV. And then I also have one for whether a gene has a 1-to-1 high confidence mouse ortholog. And then for some people, those are important to look at as well, especially if you're a mouse modeler. There's a few columns on basic gene function. I include the number of EQTL SNPs in GenCode because the median across protein coding genes is like 387. And if you think that EQTLs are going to solve the problem for us, they're as susceptible to LD as anything else. That doesn't mean they're not important. They are, but it's not as straightforward as some of us had hoped they might be. I have the mean expression of a particular gene across all GTEx tissues, whether it's a synaptic gene ontology consortium gene. This is a curated set. Is it a transcription factor gene, a housekeeping gene, or an RNA binding protein? And the definitions are all traced back specifically to these references. So the other thing I mentioned is all the stuff that I have here has a specific source. Usually it's a PubMed ID, and it allows you to go back and actually trace back exactly what was done and have a citation for it at hand. Next are some of the psychiatric genetic ones. The first is a true-false flag for whether this gene, plus or minus 10 kb in either side, has a GWAS hit in the GWAS catalog. This was updated in the current version a couple of days ago. And then if there is a GWAS hit, the GWAS catalog field has a big list of the traits that are associated. So it's a trait, / another trait, et cetera. So you get an idea as to what else is there. Similarly, for online Mendelian inheritance in man, these are often single gene hits or Mendelian disorders. A true-false for whether the gene has an entry, and then if true, OMIM list gives a string of all the hits for it. Mendelian psychiatric are from this PubMed ID, and these are inborn errors of metabolism that are known to have psychiatric manifestations. And so they're listed there. The next are whole exome findings from the ASD paper from Fu et al. In 2022, developmental delay, neurodevelopmental disorders, as well as the schema schizophrenia ones, both for the Bonferroni significance and the Q-value significance. So these are all important, I think, to the stuff that we do. There's a source called SysID, which has lists of intellectual disability genes, ID genes. They're listed here. And then these 4 columns summarize whether this particular gene is in a copy number variant associated with ASD, developmental delay, intellectual disability, and or schizophrenia. Oops, I'm sorry, this is triggered. And then finally, the main one here is this gene in the PGC 2022 Nature paper for schizophrenia? Is this gene in an LD-defined locus? So this is a fast way to get the GWAS hits. In the same paper, they came up with a list of priority genes, and those are listed here. So it's relatively easy to come up with that set of genes if you wish. Next, there's some other stuff for gene expression. The ones listed here from ASD, log2 fold change, it's FDR, and whether it's a true-false flag as to whether it's significant at 0.05. These are from Mike Gandel's paper in Science, where he did a meta-analysis of case versus control in cortical gene expression for autism, ASD, bipolar disorder, schizophrenia. And you can look at, these are fast ways to identify genes, which, at least in this paper, were found to be differentially expressed in cortex based on bulk RNA-seq. And then I've computed the mean expression across the GTEx tissues for adipose, adrenal gland, blood, but the ones generally of interest for us will be brain cortex and other. Again, this is bulk tissue RNA-seq. The caveats should be noted, but at the same time, this is a good, fast way to find out in which tissues is a particular gene found. Now, so let's do something with it. So I used code basically to pick, to identify the 1676 protein coding genes that are in a schizophrenia locus from the PGC 2022 paper. And it was done with this. So I loaded the GeneMatrix and then filtered to get rid of duplicated genes, to keep only protein coding genes, and then to get the ones that are in the locus, and there were 1676. Then I picked the subset that were also of these that were also implicated by the whole exome sequencing study from Schema. And then you get a data frame as I show here. Let me make this larger. And so this is the ensemble gene IDs. These are the actual typical gene names. And then you can find a whole host of information about these. So for example, many of these are highly conserved, as you would expect. PLI close to 1 for 4 out of the 5. All of these have additional GWAS hits, including schizophrenia. Several, 3 of 5, have an OMIM association. And if we scroll over, we'll see that a bunch of these are ID genes, and quite a few, 3 out of the 5, are actually neurodevelopmental disorder genes. So this is the kind of stuff where if you're trying to understand your findings, there's a fast way to actually get at this. And then I just did a bunch of other crosstabs that 47 of the whole 47 are also neurodevelopmental delay genes from whole exome. 137 are also ID genes, synaptic genes, transcription factors, et cetera, et cetera. So again, with some simple code, this can actually make a lot of your bioinformatic life a lot easier. This is a way to come up with a short list of genes that are defensively important in schizophrenia. So here, I begin with a gene matrix. I make a data frame called SCZ. Protein coding, I keep autosomal genes by getting rid of ones that are in XY in the pseudoautosomal regions or on the mitochondrial chromosome. And then I pick the ones that are either whole exome, a priority gene, a Mendelian psychiatric, or are in a C and V associated with schizophrenia. And in the end, there's 502. And here, just a bit of code. I basically made a table comparing gene size, coding region size, constraint, PLI, and brain cortical expression. These are the genes that are not in the list. These are the rest of the genes that are. And then as you can see, the genes implicated for schizophrenia are far larger than those that are not. Coding regions a lot bigger, somewhat more constrained, and have higher levels of expression. Okay, that's it. And happy to take questions afterward.
Thank you, Pat. What a fantastic resource. I'm sure that would be really valuable to lots of people listening online. But let's move rapidly on to the very third talk of this afternoon. And I'm sorry, Mike, I've just lost your title. That was me stalling there. So it's a real pleasure to have our third talk from Michael Love, who is in the genetics department at UNC-Chapel Hill. And his talk's going to be on Tidyomics. I don't know if I've even said that right, Mike. I'm sure you will get it right, which is a framework for exploratory data of exploratory analyses of genomic data. So I'm going to hand over to Mike, who will do much better than I have here.
No, that was perfect. That was perfect. Thanks so much. And this is a really great venue. So I'm excited to present here. And it works perfectly going after Pat because I do a lot of this type of data analysis with Pat, and his examples in his talk lead nicely into this. So I put together this, I'm going to just walk through kind of an analysis script. I put this together for this group. So just at the very top, like the task, we're going to try to figure out if type 1 diabetes GWAS loci are near in the genome to CD4 plus cell accessibility peaks. And so I am going to, through this, I'm going to show examples of code from this large project. It's an open bioinformatics project called Tidyomics. And so many people are familiar with Tidy, and this is like Tidy analysis and data applied in the omics context. And so you can go here. This is like github.com/tidyomics, and we've got a lot of information here. I want to point out it's a large project. There's dozens of developers working on this. And if you want to get involved, there's links here. And most importantly, I want to point out you can always come to this Slack channel in the Bioconductor Slack and just ask questions. There's a bunch of people there who are eager to help out. So I'm gonna walk through this in my time, this script here, and I'll point out this is online. And maybe I can provide the links later so that it's accessible on the YouTube. It's basically CD4 hyphen overlaps, and then it's a subdirectory where we're going to look at the C atlas, and then we're going to be walking through this script here, so you can go and try this out on your side as well. So just to mention the data sources, I've downloaded bed file locations of accessibility of T1 lymphocyte to CD4 positive cells from the C atlas, which is a really nice resource, single cell atlas of chromatin accessibility in human genome. And I've also downloaded already type 1 diabetes GWAS loci from the GWAS catalog. And then I also have some special file, and I have to figure out if Pat is willing to also put this on, because it's kind of like the extension of GeneMatrix for the entire genome. And so Pat has put, we've been working together on some projects, and he's put together this file, which has a lot of convenient metadata on the genome in 1 KB bins. And so I'll go very quickly through kind of some setup because it's not that as interesting. Basically, here are my files. This is called, the bin data is the genome in 1 KB bins. I've got the GWAS, and then these are the CCRE, that's the candidate cis regulatory elements as detected with single cell attack. Okay, so I read it in, and I do a little processing. You can read this over on your own time. This gives me the genome bin data. I read in the GWAS summary statistics. I do a little processing. And then I'm going to move into now working with the range data. So the main package I'm going to work with here is called Ply Ranges, and it's basically dplyr for genomic range data. This is a paper describing the package. I'm not involved with the initial paper, but I have become like an evangelist for this package. I've written some workflows with the original authors, and I've started to extend the software. So this package is part of a larger project called Tidyomics, and so there's other packages that are also available. And basically, the idea is it's an abstraction layer between the user and these large and sometimes complex biological data objects. And so we don't actually rewrite any data objects, or we don't write any new methods per se, but we allow people to use what we say are like familiar verbs. For anybody who's familiar with dplyr or tidy analysis, these verbs, people often know or have used these before with tabular data, and we let you use those verbs on genomic objects like from Bioconductor. And we then pipe between different operations using, this pipe or the other one, the one with the percents. And so what that means is, if you do X pipe F pipe G with an argument, that's the same as nesting it in this way. And so PlyRanges defines a lot of convenience functions for reading in genomic data. And I just mentioned the PlyRanges is just one of these packages. There's also ones that provide interfaces to gene expression matrices or single cell data or cytometry, all kinds of different packages here. Also chromatin conformation and interaction data. So we load up the PlyRanges package. And these are everything I'm showing here is on the Bioconductor project. So can be installed through the Bioconductor installation script. And right from the beginning, it's really important to identify the genome. I know from reading the documentation that this is HD38 from the C-Atlas. The bin data from Pat is HD38. And then the GWAS data, the genome is GRCH38. So we've got the same coordinate system, but as you all might be familiar, the names of the chromosomes are different between, say, like the UCSC style and the GRCH style. So there's a nice converter function in Bioconductor that lets you just change the style of the chromosome names. So that switches us over now to this like UCSC style, and then we can compare across these different range data. So the CRE, the cis-regulatory elements are these peaks, and they have some metadata like a score and a p-value or a q-value. And we can start working with it like as if it were a table. So we can say select a column, and that you always get back your core information. So like the chromosome and the strand and the extent in the genome, and then you're selecting just on these metadata columns. So now we have same data, same ranges, but now we have selected just a single metadata column, and we can start doing something. So maybe the first thing we could do is ask which of the GWAS loci are overlapping the MHC. So we just need to this is a very rough definition of the MHC from 29 megabases to 33 megabases on chromosome 6. And we can start doing some stats. So we have 566 GWAS loci. We can do a quick filter. So we're just pass this through. There are lots of options here. You can, restrict that the strand is the same. Many other options you can look up with these functions, but that's a quick task. So we get that 44 of these ranges of the 566 are overlapping the MHC. And then the PlyRanges package conceives of doing kind of intersections or asking for overlaps as database joins. So if you're familiar with dplyr, you're maybe already familiar with, the way that they implement classic database joins. So we, take the GWAS ranges and we say we want to join by genomic overlap, and we're going to do a left join. Left join means that we will get back all of the things from the left, so the GWAS ranges, including those that don't overlap the regulatory elements. So, and the way to know if we did not overlap a regulatory element would be that there's an NA instead of a value. So this means that the first range didn't have a cis-regulatory element within 10 kb from the GWAS locus. And this is the GWAS locus, and this is just information about the overlaps. If it overlaps multiple times, so you can see like the bottom 4 are the same SNP, you'll get metadata from all the overlaps. So that there are four cis-regulatory elements within 10 kb of this GWAS SNP. We can do an inner join, and that gets rid of the any of the GWAS loci that didn't have a cis-regulatory element within 10 kb. So that tells us that we have 750 hits. So there's some, obviously some double hits because we have 566 GWAS loci altogether. And then we can do some processing. So we can do a join. We can pipe that to filter. So let's get rid of these duplicates. So we get rid of the duplicated SNPs, and then we could do some summarization. So say, how often did we have so we'll just use the score to tell us about whether we had a cis-regulatory element nearby, and we find out that we have 226 unique GWAS loci that had a cis-regulatory element nearby. I'm going to go really quickly over this. There's another Bioconductor package that lets you plot range data very easily. And so you can do classic Manhattan plots, or like this is now coloring the point by whether or not there was a cis-regulatory element. And you can check that out at this link, Plot Gardener. Okay, so then the last bit, this is something that also Pat and I have done quite a bit in our different projects together, is we found that there are some overlaps, like we saw 700 some odd overlaps. We want to know, well, okay, what would we expect? And so what would we expect is often asking, what do we expect under the null? And you have to define, what is the null? There's lots of complex correlation structure of the genome and of features in the genome. There's a lot of relationships between variables. And so we have a package in Bioconductor called null ranges that allows you to specify different types of null sets or regions, particularly for doing enrichment analysis. And so at a very high level, you can read more at the package website, but at a very high level, there's covariate matching or bootstrapping are kind of two approaches. And I'm going to show you an example of doing covariate matching very briefly. So we take Pat's Ben data, and I'm just going to convert one of the variables to the log 10. So this is the log 10 of the summed up LD score in a 10 kb bin. And I can add that to my GWAS data. So now I have my GWAS loci, but I also have the log LD score of the bin that it resides in, the 10 kb or the 1 kb bin that it resides in. And you can just quickly plot. Obviously, we see this like higher LD score on chromosome 6 coming from the MHC. That's just doing a quick exploratory plot. And we can use this, we can define a pool. So let's take the whole genome, remove any of the bins that overlap a GWAS locus or hit a lead SNP actually. So this is filtered by non-overlaps, and I actually remove any bin that's within 10 kb from a lead GWAS SNP. And then we're just going to do a little downsampling. So we get 100,000 of those, and we pipe that into a function called match ranges. Match ranges, we say, these are our SNPs of interest, or our ranges of interest. This is a pool of other regions. And would you please match on these two covariates? So GC content and log LD score. And that gives you back a match set. You can see that plots for the different covariates. So here's the GC. Here's the log LD score. Can't exactly match. These are the MHC ranges, and it's very hard to exactly match on those, but we do get closer than with the whole pool. And then we can finish off by putting together the GWAS SNPs and the matched SNPs from the rest of the genome, and we can compare, do we see more overlap with the cis-regulatory elements from the GWAS SNPs or their matched set? And we see a little bit more, so well, 3 times more here, including duplicates for the GWAS. So I'm going to wrap up there and take questions in the back.
Hey, thank you very much, Mike. It's a tour de force, and I really love the way these 3 talks, although in quite different areas, have really moved us towards making really difficult analyses, difficult interpretations, much more straightforward. And I think that's exactly the direction we need to go into in the complexity of everything that we're dealing with, particularly for psychiatric traits. So let us move on to questions. We have 15 minutes here. I'm going to start with the 2 that are already in the Q&A panel, which I assume the attendees can see, as well as those of us that are on the webinar panel. So, Ollie, one for you, asking about whether the GenoPred pipeline can work with methodologies, polygenic score packages outside those that you list on your diagram.
Yeah, so good question. The pipeline does implement PRSCS. It's just not in that picture. I mean, users can potentially work with me to kind of build in other methods, but I would say you can always use these other methods. One thing I didn't mention is that apart from providing GWAS summary statistics, you can also provide previously derived score files. So if you have score files that you've generated using another method and you want to plug that into GenoPred to then calculate the scores in your target sample in a kind of ancestry, adjusted way, then you can do that. Similarly, if there's existing score files on the PGC catalog, the polygenic score catalog, you can also just download those and use those as the input.
Thank you. And while you've got the floor, maybe I can just ask the 3rd question on there, which is also for you, and then I'll come back to Patrick. Can the polygenic score pipeline handle multi-ancestry data sets?
So good question. It assumes the GWAS summary statistics have been derived from a single ancestry. If they haven't, then we recommend users to specify the population that represents the largest proportion of that GWAS sample. There is future work planned to incorporate methods that allow GWAS from multiple ancestries to be used in kind of polygenic scoring methods such as PRSCSX and Bridge PRS. In terms of multi-ancestry target samples, yes, there's no issue there at all. It will take those individuals, it'll estimate which populations they match in the reference population, and then scale and standardize the polygenic scores according to that ancestry matched reference.
Thank you. Clear. Pat, I'll come to you. Did you also want to make a comment on the polygenic score ancestry, or was that a no? Okay, then I'll hand over to you for the question at the top about gene.
Yeah. So Julie White asks, is GeneMatrix built programmatically? And the answer is yes, it is. But it's got 2 basic components. The first is essentially probably the one that has to be updated most often is the reading a particular file from GenCode and parsing it to get the gene information out of it. Then there's part of it that goes through Biomart to get some information as well. And that part is updatable and programmatic. The other things I add, which are essentially findings from studies, those are not readily, I mean, you can go and find the table and process it yourself if you would like and quality control it, which is what I do. But that ends up being about 20 different scripts. So I guess the answer is the core one to build the key identifier lists. Those are things which certainly could be shared. And if there's interest, I'll try to get it on the PGC GitHub. And that sounds like there is interest. So I will try to do that.
Great, thank you. And just those of you that haven't seen, Pat has uploaded to the webinar chat links to his R code and his PowerPoint, so do check that out. And of course, as always, this seminar will be on the PGC YouTube channel very shortly. So is there anyone that wants to ask a question in person? And other written questions are also welcome in the Q&A. But maybe while people are thinking what else they might want to ask, maybe I can just ask each of you, what's next? You know, what's the next developments that we're going to need to take this area further, and what are your what would be your wish list and what you're going to do? And Mike, maybe I start with you since we haven't heard from you in the Q&A session.
That's a good question. I don't have anything prepared. I mean, I'll just say, okay, I'll say two things quickly. One is, I think the reason I've invested time in this like pipeline, you know, basically everything you saw today is, I think exploratory analysis is so key. It's easy to compute p-values. It's hard to figure out if you've made a mistake and something is wrong version, confounded, wrong files. I think like it's so important. And then the other thing I'll say, just because people are here, I very appreciate the openness of the PGC. It's fantastic. You're, you know, this is like the way to do team science, and I really appreciate the people, whoever is out there providing the conditional signals for the GWAS. This is something we care a lot about, and I appreciate that you take the time to do that, because not every disease-focused GWAS consortium is onto conditional signals.
Oh, right, interesting. Thank you, and I'm really glad that works at the PGC. But this is very much a partnership, and it's great to have you here today. Pat, what's next?
Yeah, a better annotation. That is the summary. Right now, we have just the vaguest idea of what most things in the genome do. At the gene level, certainly at the transcript level, because the transcripts of a gene are poorly cataloged, and they're not brothers and sisters. They're distant cousins or even completely unrelated, sometimes with exactly opposite functions. So the same gene can make two transcripts, one that increases something, the other that decreases something. And we consider them as being equivalent, and that's almost certainly incorrect. In addition, especially for whole exome and certainly whole genome analysis, annotating variants is the Achilles heel. We don't know with confidence what most missense variants actually do in whole exome, and that's a huge weakness for us. So annotation.
Good annotations, thank you. And Ollie?
So future plans, as I mentioned, one to incorporate methods that can handle multi-ancestry GWAS. Another area I'm really interested is the integration of functional genomic annotations with polygenic scores. I'm very interested in trying to compartmentalize genetic risk into biologically meaningful kind of components, and we're already doing a few things on that front. And then for further work also, I'm just kind of improving the interpretability of the scores, so that when you get one, you can actually understand what that means in terms of that individual's risk. And yeah, that's it from me.
And there's one more question in the Q&A for you, Pat, from Mark Adams, about formatting results from GWAS papers so that gets easy integration with GeneMatrix. You're muted, Pat. Pat, you're...
Sorry, that's a great question, Mark. Yeah, we should look at that. We have been doing a bit of work. Lu Yi and I have come up with a way to make sort of a VCF-like GWAS SumStats format, which so like currently everything's in the name, right? Which is not the best way to encode the definition of fields, the definition of where things come, what the model was. We have a rich, like a VCF-like header with all the details, what the model was, where the cohorts came from, how many cohorts, when it was done, what build it was done, who did it, all that. That would help some. And in terms of other stuff, I think that's a great one for the community to think through. Like exactly how can we come up with sort of a standard summary or a standard list of loci, for instance, that can be used, because that's really kind of the key step, I think. So great question, Mark. I don't have a great answer.
And a question from Frida Lola, asking you, Pat, about protein coding genes.
Yeah. So I'll mention that the GeneMatrix does have all 38 biotypes. There's 62 or 63,000 entries, and link RNAs are there, microRNAs are there, knock yourself out. It's just that when I do my own analyses, I typically focus on the subset that are protein coding. And the reason why is because, I mean, I spent like a week trying to find a database of link RNAs that were known to actually do something, and I wasn't able to find it. If somebody knows, please let me know, but I looked futilely for a long time. A lot of these things are essentially predictions, so in silico predictions, and many of them, in fact, are not real. A bunch are listed as novel, but there's little evidence that they're actually ever expressed anywhere. In addition, this is incomplete. There's a number of reasons why this is incomplete. In addition, there are areas of the genome that contain, the genome is not one thing. The whole concept of branching out to the pan-genome, which is a much more complicated way to accurately represent the variation in the human genomes, is something that's super important as well. That'll be something that'll eventually come up. When I focus on protein coding genes, it's because there's good evidence that they actually make a protein somewhere, somehow. So that's why. But that's really just my acknowledgement of the limitations of annotation. And all the GenCode data are there. Go for it. Use it.
And on that note, at 3 minutes to the hour, maybe we'll wrap up there. I'm sure the speakers are very willing to engage further by email. And again, in the chat, there's lots of links that you can follow up on the talks from today. So it just remains for me to say a huge thank you from me and all our attendees. We had 100 people live, which is great. And to thank our speakers, so Michael Love and Patrick Sullivan from University of North Carolina, Oliver Payne from King's College London. And maybe I'll just finish with Mike's quote, which I really like, of it's easy to figure out p-values. It's hard to figure out if you've made a mistake. And I think we would all, that puts it so nicely. And I think we've heard, 3 fabulous tools that will enable us to make fewer mistakes and find them more easily and do good reproducible science. So thank you, all 3 of you. And just to finish and say, whether you're online now or listening on YouTube, if you have ideas for a worldwide lab meeting, we'd love to hear from you. Just drop me an email. We'll finish there. Thank you all very much.